Hello and welcome to this third video showcase of the virtual brain. Today we will focus on how to simulate epileptic seizures in a large-scale brain model. Therefore, we will follow along this article called The Virtual Epileptic Patient. What the authors in this article did, they recorded empirical data from a patient with temporal lobe epilepsy. The empirical data in this case was stereotactical EEG, so they implanted electrodes into the cerebral cortex of this patient. They were able to record electrical activity of a simple seizure, up here where those fast oscillations can be seen in just one region, or in the region close to just one electrode. But also they were able to record complex seizures, where there's fast oscillations and activity in the regions of many different electrodes. Also, they were able to apply an electric stimulus into one electrode and trigger a seizure this way. So these authors were able to recreate this data in their simulation of a brain network model. Now we will look at how this can be done with the virtual brain. So first of all, as always, we need structural connectivity to start our simulation. This is the same thing as the authors did in this article. So they scanned this patient in an MRI scan and calculated the weight and track length matrix, thereby generating the structural connectivity. This connectivity was then uploaded into the virtual brain, and each region of their network model was equipped with the Epileptor. So we've discussed this Epileptor model before, but one parameter we haven't talked about is this very important parameter called XO. And I highlighted part of the text here. So this parameter XO controls the tissue excitability and is epileptogenic, triggering seizures autonomously if XO is greater than a critical value of minus 2.05, otherwise the tissue is healthy. So this means by varying this XO, we can set this Epileptor model to trigger seizures or to remain stable. So we now can create different configurations of this local model and equip them to different regions in our network. This is shown here in figure 1, where, for example, this red regions or this red spheres called the epileptogenic zone has the configuration of this Epileptor model with a very high excitability value. Here they write XO was set greater than the critical value plus 0.4. And those blue regions representing the propagation zone have just an elevated excitability of between the critical value and the critical value plus 0.4. Finally, all the other regions, these white regions, have an epileptogenicity parameter XO below the critical threshold. So we generate different, as we said, we generate different configurations of this Epileptor model and equip them to different regions of our network. So in this article, or in our tutorial now, we will follow the clinical estimation of epileptogenicity. This can be seen here in this figure 6. So the clinician looked at the SEEG data and other recorded data and predicted the epileptogenic zone, so the area in the brain which was responsible for triggering those seizures. And they found those bright colors here are the areas with very high epileptogenicity, and those red colors area are the propagation zone. So we will try to approximate this in our TVB model. Now let's switch to TVB. We're here on the faceplane viewer. As we can see, we've discussed the Epileptor model before, and now we want to create different configurations. So here's this XO parameter as discussed, and we can manipulate by moving this slider bar, and then we set very high values of XO, create epileptogenic configuration. We hit the save button. Then let's put one very close to the critical value, say minus 1.9. We call this the propagation zone. We will save this configuration as well by hitting the save button on the right side. And finally, we put one value well below the critical threshold, and call this all the other regions, and save this configuration again. Now we switch back to the simulator page, and here we can find the button called Set up a region model. And here we can give different brain areas different configuration of our local model. So in this drop-down menu we can choose our configurations, and we use the epileptogenic. Let's set this to the amygdala, the hippocampus, and the parahippocampal region. Apply to selected nodes. Then let's set the propagation zone to be part of the temporal cortex. And finally, we give all other regions the stable configuration of the Epileptor model. Finally, we hit this submit button, and we switch back to the simulator page, and we would set some other parameters like the simulation length, conduction speed, and so on, and finally hit the play button. And in the next video we will see our results and compare them to the results in the article. Thank you for watching.